So thank you all for hanging in there. I'm the last one before break, so we'll try to make this quick. Imagine that from a scoop of water, we could identify all of our species in the aquatic environment that we're interested in. Today I'm going to be presenting results on a proof of concept study where we're trying to do just that. It's a technology called EDNA. It stands for environmental DNA. And essentially what we're doing is we're trying to identify our focal species from the environment itself, and in this case, the water. There are many people with this work that I'd like to thank. I have funding partners, crews that have been helping collect samples in the field, as well as collaborators. So I thank many of you are here in the room, and thank you for your work in helping me complete this. Today I'm going to be talking about how we can use eDNA to understand aquatic biodiversity in our streams. So first I'm going to present a proof of concept study, and then I'm going to talk about a broader scale study across the whole Pacific Northwest, so the west side of Washington, Oregon, and Northern California, and that's where our sampling and OESF comes in. Worldwide, our freshwater environments, the biodiversity in our freshwater systems is among the most imperiled. Essentially, when you compare our freshwater environments to our terrestrial and our marine ecosystems, we have the highest number of listed species, so those that are sensitive, threatened, or endangered. Traditional sampling in our aquatic environments is based on visual detections and often includes counting of the species that we find, which is not always standardized across crews and across regions. And it's also dependent on the practical and taxonomic expertise of the individuals that are doing those samples within the field. And it's often been limited to assessments of adults. And in general, it's also limited to, because of funding, to those species that are listed. So these species that are actually threatened or endangered in our systems. Sampling generally has occurred in summer due to safety issues and our inability to access a lot of our aquatic environments at different times of the year. And so because of this, we've had a limited understanding of our year-round biodiversity in our streams, as well as for different life stages of our aquatic critters. This is an example of fish distributions along a coastal stream here in Washington. And what I want you to focus on is where our sampling in those streams generally occurs. So where I've circled here is in our systems where we generally have our coho and our steelhead that are listed, and that's in general in our streams when you put them all together where our main sampling is occurring. It's occurring in these mainstream locations. We have less understanding of our fishes that are up, further up in the network, as well as other critters that are swimming alongside our steelhead and our coho. So, I've sort of created or explained the challenges to understanding aquatic biodiversities in these systems. And since 2012, there's been a new approach out called environmental or EDNA Metabarcoding. And essentially with the new EDNA Metabarcoding, we have the ability to understand more about biodiversity. So what this study here shows is that more fish species were detected using EDNA Metabarcoding than traditional samples. So if you note on the y-axis of this graph, you have the different aquatic systems. So you have a control system, a pond, then ditches, lakes, streams, and rivers. And what you will note is in all those different types of aquatic systems that the eDNA Metabarcoding results match or exceed the number of species that were found in those traditional surveys that were done in each of those environments. This eDNA Metabarcoding approach uses one gene, and what it does essentially is it's a ray-finned boatyfish gene. So essentially all the fishes that would be our ray-finned boatyfishes have sequences that come out from this result, and it's from those sequences that you then identify, you map those sequences to our known understanding of what those sequences line up to the name of the fish. So we'd get a sequence out, we'd say that's coastal cutthroat trout, another sequence out, we'd say that was rainbow trout. And so your ability in using this approach to do the metabarcoding is only as good as the genetic information that's available for each of those species. So some of our species that don't have genetic information available will essentially come back as For example, it could be group C for the Onchrinchus. I know it's something related to the other ones, but I don't necessarily know what it is. It's sort of an unknown Onchrinchus species is how one would come out. We have found that we get a lot more of those for example, in our system, sculpins that tend to be more cryptic, and there's just at this point less genetic information available. We get a lot more of those sequences coming back less known. So our goal, building on that 2016 metabarcoding approach, was to develop a new approach that would be able to be used in this region that would allow us to gain species-specific aquatic data. One of the issues with that general barcoding approach that I just showed there is that when you're sampling in the systems, you have an inability to identify rare species when really common ones are found in the water. And essentially the common ones sort of blow out your ability to detect those less common sequences. And so what we want to be able to do is to have a rapid and accurate assessment of our stream environment where we will be able to eventually understand presence, potentially relative abundance, as well as the diversity of multiple taxa. So our fishes, our amphibians, our crayfishes, our macroinvertebrates, our mussels, especially during time periods when traditionally we haven't been able to sample and or at different life stages that we haven't been able to yet capture. And we wanted to be able to identify common as well as those rare species that in the previous metabarcoding approach wasn't obtainable, as well as cryptic and endangered species. You could also add invasive species to that list. So what we're developing at the Pacific Northwest Research Station is taking the EDNA Metabarcoding approach, and we're coupling it with taxon-specific primers that will allow us to identify several species of interest across multiple aquatic taxa. So what we do is we take a sample of water in the field and we filter the water, and so you'll see next to that water sample two different filters. One filters from a system that has more sediment in it than the other system. Those samples take more time to filter also in the field. And then after the time, so essentially we collect the water in the field, but we leave the field with a vial and a filter inside our vial, and we freeze our samples, and so we bring that back, and the next step is the DNA extraction in the lab, and that's just a bunch of, what it looks like is a bunch of fancy chemicals that sort of go over the filter and allows us to remove the DNA. And the next step from there are two different machines that we process our samples through. So we do an amplification and then a sequencing. And so we're amplifying the species at which we're targeting. So we are again using those metabarcodes as well as some taxon-specific primers. And the idea is that then in the end, we have this stream assemblage profile of the different sequences that we got asked for, and we're able to get the detection and estimation of relative abundance. So our proof of study concept was done on Fall Creek on the Alsea Basin in Oregon, and we wanted to just answer really general questions to understand how this approach would compare to what we expected. So we wanted to understand whether we could actually detect multiple target species across the diverse taxa. So essentially, can we detect fish as well as we can detect amphibians and as well as we can detect crayfishes? Can we also detect our common species and the rare ones in our system? Because we're interested in being able to identify both of those, so not just our common species. And can we track the change in the aquatic assemblage that moves upstream that we know occurs based on our traditional samples? Can we track that and find similar results using eDNA that we would expect? And so these three questions we really tried to get at with this study. These are just an example of our metabarcodes and the taxon-specific primers that we use. So again, we're going to be looking at fish, amphibians, crayfish, and a variety or a few different animal and forest pathogens. So first, just focus on the left on the map that you see here. So again, this is from Fall Creek, which flows into the Alsea Basin. I want you to note that down below that dotted line, I have 3 points there. 2 of them are downstream of the hatchery outflow on that system, and 1 of them is actually the water that came out of the hatchery outflow. The reason why we chose this location as our proof of concept study is because the hatchery was located there, so only fishes that are passing above are those that are native to the system, or I'm sorry, are wild to the system. And so we would see an actual break in what we would expect in the assemblage or the distribution in the stream environment, which is why we chose there. And then we have 2 points above the hatchery location, so one a few hundred meters above the hatchery, and then one further up in the headwaters, but still within the fish distribution within the network. So again, there's 5 different locations that I'm going to be presenting these results from, and I want you now to look on the right side. So the bars, the grouped bars line up to those different sites, the data for that different site. And so coho is noted there in yellow, our rainbow or our micus steelhead are there in blue, and our cutthroat is the red or brick colored. And so what you see here is that cutthroat trout are mismapped using that taxon general primer. So again, we are using a combination of the Metabarcode, those taxon general primers, and we're coupling it with taxon specific primers for these results. If we had not yet considered the taxon specific primers and we were only using this approach, we would be mismatching cutthroat trout at the outflow of the hatchery, because that's it's a hatchery that raises rainbow trout. So they have raceways filled with rainbow trout at this location. Don't actually have cutthroat in the raceways. They do, of course, have them upstream, and the water it comes in from the stream into the hatchery and then flows out back into the stream itself. So we would imagine that we would get hits for cutthroat, but not at the level that we see here. And so what we see, again, is what we expect and what papers, the few papers that have been done on Metabarcode showed that essentially when you have sometimes really common species in the system, you can mismap some of your less common sequences at those sites. And we see that here. And just to show a little bit about why that can happen and how that happens, this again is that ray fin bony fish are taxon general primer alignment. And so what you see here are rainbow trout and then cutthroat trout and coho in order. And I just want you to notice that the first 2 lines of the rainbow and the cutthroat trout are similar up to the one difference right there. Essentially, there's a 0.4% divergence between the 2. And so you could see potentially how with this taxon general primer, they could be mismatched. Because essentially what you're doing with eDNA results is you have fragments of DNA that were on that filter that were then extracted and that you're looking at. And you could see how potentially fragments could break and or could be misread with this primer set. Coho are more different than the other 2 and are less likely to mismap in this circumstances, but again, they're more similar than other fish taxa. So now looking at some of the taxon specific primers that we developed, again looking at our coho, our rainbow, and our cutthroat, what we see here is what we would actually have expected to see in the stream. And so I just want you to first focus on the rainbow trout, the blue color. And so what you essentially see here is that the highest level of our rainbow trout based on the map reads per species at the outflow of our hatchery, which is what we would expect. I also do want to say is that our results at this point with eDNA, species-specific eDNA on a single species does a really good job for presence and absence. And right now the research is sort of pushing the envelope and trying to get the ability to potentially say something about relative abundance. And as you see, the results that I've been showing you here are about the mapped reads that we're seeing, essentially the mapped reads from our sequences that map back to those species. And so we also think that we might be able to say something about relative abundance with this approach, but it's something that we're trying to get into yet. The reason why I bring that up is because if I was to only present this data as presence-absence data, it would look a little bit different and we would see presence at all of these sites. And so what the data that I'm presenting here for you today is to sort of push that envelope on where we are really with the research and what we can really say about, or potentially say about these species. So now I want to try to get into the question of how the assemblage potentially changes as we move upstream. And so what you see here are the x-axis has downstream locations located on the left-hand side. And again, they're moving upstream, those 5 different locations, with the middle location as the hatchery outflow. And so the bottom graph is our micus, our rainbow trout, or our steelhead. And that second graph up is the cutthroat trout. So what you notice is here is that I have the 3 different primers that we developed. I have our taxon general primers and then our taxon specific primers here. And in general, we see what we would have expected to find moving upstream, and that we have the highest number of our reeds from our rainbow trout steelhead at the hatchery outflow, and those that are above that line were only those that were passed above, or that's our expectation. And so based on looking at our Pacific trout here, we see that taxon-specific primers are warranted to essentially cross-validate the taxon general primers that are used in environmental DNA. And up until this point, they haven't actually been put together yet in a study, but most of the studies that have done these metabarcodes essentially say we need to have these taxon specific primers to allow us to get at those rare sequences, and what we show here is that it works and it is necessary. So looking at results from our coho and our Chinook salmon, very few of those individuals are actually wild in this system and passed above, potentially shown here. And then our last group of our fishes that we've looked at right now in the system are our sculpins. And it's just an example of where the taxon general primers were actually really useful in this circumstance because there isn't as much genetic information available on our sculpins. And so when the data is limited, those taxon general primers can actually be really useful. In addition, at this point, we're getting results back. And so we have results on our minnows and other fishes in those systems that we don't have yet taxon-specific primers back, but we're able to say something about those groups because of that taxon general primer. And then again, just sort of looking at that same data for the macroinvertebrates that we looked at in the system. So the crayfish at the top, mayfly and stonefly at the 2 lower location or the 2 lower panels. And what you see here is that crayfish essentially increase. We're showing at least an increase in their relative abundance, if that's what we're seeing here at all, as you'd move upstream. And lower reeds at the outflow for our mayfly and stonefly, as we'd anticipate, because they should be lower at sort of a more disturbed location. So our take-home messages from what we've seen here are that there's utility for using EDNA metabarcoding coupled with those taxon-specific primers that will allow us to actually identify species from multiple taxa, and that we can track changes in the aquatic biodiversity across the stream continuum, so as we would have expected. And that using multiple primer sets provides complementary views of a species and or a common ecosystem. Essentially, it allows us to have built-in redundancy. So it allows us, depending on how many genes or primer sets we were to develop, but for our Pacific trout, as an example, we have 3. So we have 3 different places that we can look for the presence or absence of that species, which allows us to say a lot more about false positives and have more information to deal with false positives from. And that the multiple primer sets actually allow us to focus on different subsets of the taxa within our aquatic communities, and that's necessary to sample the entire aquatic community in a reasonably comprehensive way. And so we feel like it's a combination of putting it all together that actually provides the most complete understanding of the story of what's actually occurring in the streams. So from that first phase, we hope to actually go out again this July, and we're going to be comparing our EDNA metabarcoding approach with taxon-specific primers and electroshocking or electrofishing in those same systems, in the same sites. And so we want to do the side-by-side comparison to actually understand more about that potential for relative abundance piece. We didn't do it when we first did our sampling because we thought our results were going to be, the strength was going to be in the presence and absence. And then we see something, we might be able to say something about the relative abundance, and so we want to go back out and look at that and whether we really can. And so I should say this proof of concept study was our first study that we're working all the way through the pipeline of essentially the approaches. So we're at the point where we're digging into the data and we're developing our essentially our bioinformatics pipeline to be able to deal with all this data. Most of the studies are dealing with one gene or one taxa group. Most papers come out, we'll just do macroinvertebrates or just do fish. And of course we decided to do multiple taxa with 3 genes of each. So, and then each of those has all these sequences that come out. So there's just a lot of data that we're dealing with. And so we're trying to set up a user-friendly pipeline to be able to deal with this data. And that's the point at which we're at right now with our proof of concept study. And we have 3 other projects that are going to be sitting on top of this study. And this one is the one that I was referring to. It's the study on the west side of Washington, Oregon, and Northern California. And so the sites that are shown there in yellow are the sites that are actually on OESF land that were collected by Kyle and his crews in 2016, 14 different streams. And then the green sites are also sites that were sampled using that same exact protocol throughout the rest of the range, collected by a combination of RP&W crews and ARAMP crews. And so what we're hoping to do here is to actually link the occupancy of these species to the habitat data that's available across many of these sites, and then to understand how downstream points can integrate some of that biodiversity data upstream. So some of the sites and that are in green are actually set up so that we have 1 site downstream and 8 sites upstream in parts of that network. And so we want to understand what that relationship actually looks like. In addition, the data from OESF will be able to compare to Kyle's fish in hand data that he has from electrofishing to actually our results we'll have from EDNA, so it'll be a more enriched data set as well.